So in this lecture, we're going to talk about experimental study designs. When we're discussing experimental studies, it's helpful to have an understanding of the symbols that are used to denote different aspects of the study. It is convention to use a capital letter R to denote the random assignment of subjects to treatment groups. An X represents the application of an intervention or treatment, and an O represents an observation. Experimental studies most closely resemble controlled laboratory experiments and serve as models for the conduct of observational studies. The defining feature of an experimental study is that the investigator actually manipulates the independent variable. They apply some intervention or treatment to a group or groups and compare those groups to a control. These designs must have one or more experimental groups, a control group, and the participants must be randomly assigned to these groups. We're going to go through a few of the classic experimental designs that may be used when conducting a randomized controlled trial. Experimental designs almost always test the hypothesis that the experimental group will differ from the control group in the outcome variable of interest. The only caveat or the only exception to this particular rule is equivalency trials, in which case you're just trying to demonstrate that the second drug is as good as the first. Sometimes investigators are unable to randomly assign people to treatment conditions, but still wish to apply an intervention to one group and compare it to a group without the intervention. Quasi-experimental designs have most of the necessary features of true experimental designs, with the exception of random assignment to treatment conditions. Now, the general form of an experimental study design begins with a research question from which a hypothesis is developed. Study subjects are then recruited based on specified criteria, and informed consent is sought for potential subjects. Eligible and willing subjects are then randomly allocated to receive one of the two or more treatments or interventions being compared. Study groups are then monitored for the outcome of the study. This could be recurrence of disease, first occurrence of disease, it could be getting better, or it could be side effects. Finally, the rates of the outcome of interest are compared across treatment conditions. The reference population refers to the general group to whom results of a trial should be applicable. The study or experimental population refers to people who are considered for enrollment in a trial, so the potential participants. We can look at the people enrolled in an experimental study through a population hierarchy. We start with a reference population, which is everyone to whom the results should be applicable. From there we get the experimental population, everyone who is actually a potential study participant. From the experimental population we have those who do not participate and those that actually make it into our studies, who are the participants. We then take those participants and randomly allocate them to treatment and comparison groups. Each treatment and comparison group can be further subdivided into those that cooperated and completed the study and those who did not. In experimental studies, you must first consider the size of the study. This does not just mean the number of people in the trial, but also the number of outcomes you expect to examine in the trial. Generally speaking, the more outcomes you want to consider, the larger your study will need to be, meaning that you will need a larger study sample. You must also consider restrictions on who is eligible for your study. You need to ask yourself the substantive questions: what groups am I interested in, and who exactly do I want to study? You then must consider the logistics. What group is actually accessible to you? Who will comply with the study protocol? How feasible is it to get complete and accurate follow-up on all the subjects? Finally, you need to consider the characteristics of the volunteers. How does your study population differ from the total experimental population? Are your results going to be generalizable to the population at large? Now, in the pre-test, post-test control group design, participants are randomized into either the experimental group or the control group. Both groups are then measured before the introduction of the intervention. An intervention is then applied to the experimental group, and finally both groups are measured again. Random assignment ensures that the groups are roughly equivalent on important attributes at the start of the study, and helps to control for most threats to internal validity. Now, the post-test only control group design is especially relevant when we think that the pre-test might influence the outcome of the post-test, or when pre-testing is impractical or unfeasible. This design assumes that the random assignment will create equivalent groups, but does not evaluate that assumption. In the post-test only control group design, participants are randomly assigned to either the experimental or control condition. The intervention is applied to the experimental condition, and then both groups are measured for the outcome. Now, if we are concerned about change between pre-test and post-test, but we are also concerned that pre-testing could influence the post-test, we can use the Solomon four-group design. In this experimental design, you randomize participants into 4 groups, 2 treatment groups and 2 control groups. 1 treatment group and 1 control group will receive the pretest, while the other treatment and control group will not. The intervention is then applied to both treatment groups. Finally, all 4 groups are tested at follow-up. By using the Solomon four-group design, we can assess change between pre- and post-test, as well as assess any testing effects that may occur. The main weakness of this particular study design is that it requires a very large sample. Now, the crossover design is the gold standard in pharmaceutical research. In a crossover design, each participant serves as his or her own control, allowing for an increase in statistical power, and therefore smaller sample sizes. Participants are randomized into 2 treatment conditions. As an example, say you are comparing a new drug you have developed, which is drug A, to an existing drug used to treat a particular condition. We're going to call that drug B. At the start of the study, participants in experimental group 1 are given drug A, which is our new drug, and participants in experimental group 2 are given drug B, the existing drug. Participants are then measured for the outcome of interest, at which point, usually after some washout period, the groups are switched, and experimental group 1 will then receive drug B, and experimental group 2 will receive drug A. Then each group is observed again. Now, the aim of virtually all research is to be able to apply the results to a larger group in the general population. To be able to generalize to the population at large, the sample must be similar to the population. Generally speaking, the participants in RCTs, or randomized controlled trials, are not similar to the general population due to the stringent inclusion criteria exercised to gain control within the study. However, the results of these trials do give us a sense of the efficacy of an intervention. Randomization to control and treatment conditions is designed to evenly distribute known and unknown characteristics between the 2 groups. It is designed to put both groups on an equal footing at the start of the study. Now, a placebo is an inert substance that appears to be the same as the active treatment. Not all experimental research uses a placebo. Sometimes experimental conditions are compared to the standard of care for a particular treatment that people would normally get associated with that condition. Blinding refers to when either the patient or both the patient and the doctor are unaware of which treatment condition the participant is in. In a single blind study, the patient is unaware if they are receiving the experimental treatment or the placebo, and in a double-blind study, neither the patient nor the doctor knows which treatment group the patient is in. Double-blind trials are considered ideal, however it is often difficult. Now, the choice of a control condition can be fraught with ethical considerations. In most cases, there should be a reasonable expectation that the control condition will prove to be as good as or better than the experimental treatment. This is why standard of care is frequently used as the control condition in studies, since it would be unethical to deny patients access to standard medical care they would receive if not participating in the study. Now, one of the fundamental tenets of a true experimental design is the random allocation of subjects to treatment groups. In random assignment, each individual has an equal chance of receiving each of the possible treatments. Random assignment should not be confused with random sampling. In experimental studies, you typically have a convenient sample of volunteers. These are not a random sample of the population at large. Rather, they're a group of volunteers that may or may not be similar to the larger population. You then take this convenient sample and randomly assign each individual to treatment groups. Now, the goal of randomization is to achieve baseline comparability between the compared groups on factors related to the outcome. In other words, randomization is designed to obtain two groups that have the same characteristics at the beginning of the study, thereby avoiding systematic bias within the study. You do not want one group that has a greater probability of experiencing the outcome than the other. The essence of good comparisons between treatments is that the compared groups are the same except for the treatment. Any group of individuals will vary in response to a treatment based upon their sex, age, overall health, severity of illness, or any factor that is relevant to the response to the treatment. The investigator will know some of these factors, but there are many unknown factors that may also be relevant. Randomization is designed to create groups with an even distribution of even the unknown characteristics. Now, placebos are used to make the treatment groups as comparable as possible, to have each group's experience be as similar as possible. Blinding indicates whether or not the subject or the subject and the researcher know whether or not a subject is receiving the active treatment or the placebo. In a single blind study, the subject does not know which treatment group they are in. However, the investigator is aware of the treatment group. In a double-blind study, neither the subject nor the investigator is aware of which treatment the subject is receiving. Blinding helps avoid bias in the ascertainment of the outcomes, while placebo allows the study to be blind. Experimental studies require active participation and cooperation of study participants to the study protocol. Deviations from the protocol will inevitably occur, usually due to side effects, illness, level of interest by the participant, and the length of follow-up time in the study. Non-compliance to the study protocol makes the compared groups look more alike, reducing the ability of the investigator to detect a difference between the groups. Some strategies to enhance compliance include picking an interested group and designing a simple, easy-to-follow protocol, maintaining frequent contact with the participants over the course of the study, and providing incentives for study participation. It is important to try to minimize loss to follow-up for all groups in the study. Differential loss to follow-up, meaning that one of the treatment groups loses more people than the others, can lead to study bias, especially if the reason for loss to follow-up was due to some aspect of the treatment. It is also important to be equally vigilant in follow-up of all compared groups. Non-uniform ascertainment of the outcome across groups can lead to bias in study results. Now, one definition of clinical trials is a research activity that involves the administration of a test regimen to humans to evaluate its efficacy and safety. So this is referring to a rigorously designed and executed experiment involving random assignment of test and control treatments. There are 5 main types of clinical trials: treatment trials, prevention trials, screening trials, diagnostic trials, and quality of life trials. The type of clinical trial indicates what the researcher is studying. It does not indicate the phase of the research or the research design that was used. Treatment trials test new treatments for a disease or condition, new combinations of treatments, or new approaches. Prevention trials are designed to find ways to prevent disease in people who have never had the disease, or to prevent a disease from returning. Sometimes these are also called prophylactic trials. Diagnostic trials are designed to find methods for diagnosing diseases in symptomatic individuals. Screening trials are designed to test the best methods for detecting certain diseases or conditions, typically in asymptomatic individuals. And quality of life trials are designed to find ways to improve comfort and quality of life in people with chronic illness. Now, there are 4 phases of clinical trials. Phase 1 trials are initial studies to determine the metabolism and pharmacologic actions of drugs in humans, side effects associated with increasing doses, and to gain early evidence of efficacy. Phase 2 trials are controlled clinical studies conducted to evaluate the efficacy of the drug for a particular indication in patients with the disease under study, and to determine short-term side effects and risks. Phase 3 trials are expanded controlled and uncontrolled trials undertaken after preliminary evidence suggests that the efficacy of the drug has been obtained. These studies are designed to gather additional information on the benefit-risk relationship of a drug. Now, phase 3 trials are what people generally think of when they hear the phrase randomized controlled trials. This is the last phase of trials required before a drug can be marketed to the public. Phase 4 trials are post-marketing studies that delineate additional information on the drug's risks, benefits, and optimal use. The most important thing to note about phase 4 trials is that they occur after the drug has been approved for use and is available to the public. These trials allow researchers to examine unforeseen consequences in the general use of drugs, and as such, these are deemed effectiveness studies, meaning they demonstrate real-world use of the drug. Now here you see just a list of advantages and disadvantages of the study designs that we have covered up to this point. It's not an entirely comprehensive list, however it does list the main points and counterpoints for each of the types of study designs that we've used.